Welcome student friends. In last lecture we have studied dividing cells. These are called meristematic cells. In this lecture we are going to learn the non-dividing cells, or these are called as permanent tissues. These permanent tissues are nothing but developed from differentiation process of derivatives, and these derivatives which are added by meristematic tissues in the growing regions. These derivatives differentiate from each other according to their position and their requirement of the plant, and these matured developed tissues we call them as a permanent tissues. Welcome all of you in this Alka's Botany channel. Please subscribe, like and share the video. I am Dr. Alka Inamdar and you are studying now the permanent tissues. I already mentioned that the plant body is composed by dividing and non-dividing cells, that is meristematic cells and permanent cells respectively. These are some characteristics of permanent tissues which are differ from the meristematic tissues. Permanent tissues are developed by differentiation process from the derivatives which are already added by the meristematic tissues. They are mature tissue and lose the meristematic activity of cell division. The permanent cells are fully differentiated. The cells are large and definite size and shape. Large vacuoles are also present inside these cells. The metabolism rate is lower. Permanent tissues may be living or dead, that means they are non-lignified or lignified. The permanent tissues in plants mainly helps in providing support to the plant body, protection of the parts, as well as in photosynthesis, which is a very important function of the plant, and conduction of water and minerals with nutrients for the photosynthesis. Now we come up to the types of permanent tissues in plant body. Permanent tissues are classified into two types. First one is a simple permanent tissue and second one is a complex permanent tissue. The simple permanent tissues are those tissues which are composed of similar cells and exhibit homogeneous in nature. Again, simple permanent tissues are classified into three main types. First one is Parenchyma, second one is Collenchyma and third one is Sclerenchyma. We will see one by one. First, Parenchyma. These cells are living or non-lignified. The cell walls are thin and cellulosic type. Cells can be oval or round in shape. Intercellular spaces are present in between those cells and they have large central vacuole and a dense cytoplasm. The parenchyma tissues are located in the soft parts of the plant body such as the cortex and pith. It mainly acts as a packing tissue which provides mechanical support and also helps in the storage of food material. Based on the basic functions, parenchyma can be further classified into Erenchyma, Chlorenchyma as well as ideoblasts. The parenchyma cells, when contains chloroplastids, the tissue is called as Chlorenchyma. These are mainly present in the leaves and other green and aerial plant pots. When in aquatic plants, such parenchyma tissues show large air spaces in the form of air chambers or air cavities. This type of special parenchyma is called Erenchyma. When parenchyma cells stores calcium oxalate crystals or raphides, then it is called ideoblasts. Second one, simple tissue is Collenchyma. The cells are living cells and have an elongated shape. The cell wall shows unevenly thickened and confined to the corners of the cell. These shows thickenings, that is also called as annular thickenings. The cell wall material consists of cellulose, pectic substances and water. The Collenchyma tissues are mainly located in the leaf stalks and hypodermis in the internal part of the stem or root. The main function it provides as a flexibility to the plant body as well as mechanical support to the plant. Third one important simple tissue is a Sclerenchyma. The cells of the Sclerenchyma tissue are dead or lignified. What is meant by dead or lignified tissues? During differentiation process, the cells gradually lose the cytoplasm and show deposition of secondary wall material, mainly lignin, and hence cell wall becomes thick, and this cell is called as lignified cell. That lignified cell considered as a dead tissue or dead cell, and which are non-lignified, these are called as living tissue. The Parenchyma and Collenchyma are living tissue, whereas Sclerenchyma is a simple tissue but it is a lignified or dead tissue. The cell wall is very thick due to deposition of secondary wall material, mainly lignin. The cells of this tissue exist in two forms, fibers and sclerites or stone cells. These fibers shows elongated cells with narrow pointed ends, and such cells exhibits in groups or patches. The sclerites relatively short cells with hard lignified relatively more thick wall, leaving small unthickened area, or it is called as a lumen, in the center. They are generally located in the leaf veins, hard coverings of the fruits and seeds, and also found in the surroundings of vascular bundle for the protection. The main function of the Sclerenchyma tissue is provide rigidity and mechanical support to the plant body. They also make the plant stiff and hard. These are the nature of Parenchyma cell tissue, Collenchyma tissue and Sclerenchyma tissue. These three are nothing but simple permanent tissues present in each and every higher plant body. Now we come up to the complex tissue. They are made up of different types of cells or elements and collectively form as a heterogeneous in nature. They are having common origin and performing different but closely related functions as a unit. That's why these are called complex permanent tissues. The cell elements may be living or dead. The important permanent tissues in vascular plants are xylem and phloem. Both these tissues are commonly known as vascular tissues as they are doing the function of conduction. First one, that is a xylem. It is a chief conducting tissue of the vascular plant, responsible for conduction of water and inorganic solutes from soil to the leaf via roots. It consists of four components: tracheids, vessels or trachea, xylem fibers, or it is also called wood fibers, and xylem parenchyma. Out of these four components, xylem parenchyma is the only non-lignified or living tissue, and remaining three are lignified or dead tissue. Hence, xylem is considered as a dead or lignified tissue. Now the first one element, that is tracheids. Tracheids are dead cells. They are elongated tube-like and tapering end walls. The walls are hard and lignified and encloses a wide empty lumen at the central position. Tracheids possesses various types of wall thickenings: annular, that is a ring-like structure; spiral thickening, which is a helical in position; scalariform thickening, it looks like ladder; and reticulate, that is a network or mesh-like. And these are pitted, The pits may be simple or bordered. The end walls are perforated by the presence of bordered pits, which permits flow of water from one cell to another cell. And the main function of tracheids is to conduct water and dissolved mineral elements. They also provide mechanical support due to the presence of hard secondary lignified walls. These are the structures of tracheids. It is elongated, and these are thickenings. First one is annular thickening. It is ring-like. Spiral, that is helical. Ladder-like, which is called as a scalariform, and mesh-like structure or reticulate structure, and bordered or simple pit, that is a pitted structure of tracheids. Now the second one is vessels or trachea. They are long tube-like structures, series of tube-like cells placed one above the other, and the end walls of the cells, both the end walls are perforated, upper as well as lower. Each cell appears circular, oval, or sometimes polygonal with a very wide lumen, with latter becomes lignified and dead. Vessels also possess the deposition of lignin in different form like annular, spiral, scalariform, and reticulate. It is a main conducting tissue which is in angiosperms. That means these are absent in gymnosperms. They translocate water and minerals from the roots to the leaves. They also provide mechanical support to the plants. The third one important component of xylem is xylem fiber, or these are also called wood fibers. The xylem fibers develop from same meristematic tissues as the other xylem cells. They have lignified secondary walls and a narrow cell lumen. They are usually longer than the tracheids of the same plant and present both in primary as well as secondary xylem. And the fourth one, last one element of the xylem is xylem parenchyma. These are living parenchymatous cells present in both primary and secondary walls. Both of them have walls and living protoplasm. The main function is the storage of food in the form of starch and fats. They also help in conduction of water and minerals. Wood parenchyma and ray parenchyma are these two types of parenchyma present in secondary xylem. The wood parenchyma is formed from fusiform cambium initials, whereas the ray parenchyma is formed from ray initials of the cambium. This xylem parenchyma is the only living material in the xylem, and remaining three are dead tissues. The second important complex tissue in plant is a phloem. It is a chief conducting tissue of vascular plants which is responsible for translocation of food material which is already prepared in leaves. It is translocated to the each and every plant part through the phloem. It consists of four elements. First one is the sieve elements, second one is the companion cells, third one is the phloem parenchyma, and fourth one is the phloem fibers or bast fibers. Out of these four, phloem fibers are only lignified or dead, and remaining three are living or non-lignified. Hence, phloem tissue is considered as a living or non-lignified complex tissue. In that first component is a sieve element. The sieve elements consist of the sieve tube and sieve plates. Sieve tubes are long tube-like bodies formed from a row of cells arranged in longitudinal series, where the end walls are perforated in sieve-like manner. The perforated end walls are called sieve plates, through which cytoplasmic connections are established between the adjacent cells. It is a most important food translocating structure. Second one component of phloem is companion cells. These cells remain associated with sieve tubes. They are thin-walled, narrow, elongated cells with a dense cytoplasm and prominent nucleus. Companion cells occur abundantly in angiosperms. They control the translocation of organic substances through sieve elements. Third one is Phloem parenchyma. They are thin-walled parenchymatous cells associated with sieve elements in phloem tissue. It is used for storage of food material. And fourth one are called bast fibers. These are also called phloem fibers, which are only thick-walled dead elements in phloem. They give mechanical strength to the soft phloem region. Thus, these are complex tissues, xylem and phloem, which are doing the conduction in plant body. So, these are also called as conducting tissues. Collectively, these tissues are meristematic and permanent. In last lecture, we have studied meristematic, and in this we are studying permanent tissues. Permanent tissues are mainly in three types: simple, complex and special. In that simple tissues, parenchyma, collenchyma and sclerenchyma, these are three simple tissues. This sclerenchyma again classified into fibers and sclerites. And complex tissues are divisible under xylem and phloem. Xylem shows 4 components and phloem shows 4 components. Xylem is considered as a dead tissue, whereas a phloem is considered as a living tissue, and these special tissues we study afterwards. Thank you very much.